Welcome to this tutorial. In this video, we will learn how to calculate coefficient of variation and least significant difference. As I have explained in the previous tutorial, analysis of variance and that is the foundation for both these calculations. In that video, we also learned how to calculate grand mean. Here we will once again calculate the grand mean and then use it to find the coefficient of variation. After that we will move on to calculate the LSD, least significant difference. The link to the Excel tutorial for performing ANOVA is available in the video description. Now first we will calculate the grand mean in order to calculate the coefficient of variation. Type is equal to, first select the cell where you want the answer. Type answer is equal to, type square root sqrt, double click on square root. Now mention the cell. This is error mean square, that is error mean square, close the bracket and close this whole entry. In order to represent this as one value, divide by oblique, divide by grand mean. Here we will calculate the grand mean later on. Mention the cell first, grand mean. Now close the bracket, multiply by 100, type star and then 100. Okay, now press enter. Divisor is missing, so we will first and then now we will calculate the grand mean is equal to average. Double click on average, mention the cells. We will make the selections. All six genotypes means are selected now. Now press the enter. You have seen as we calculated the grand mean, the coefficient of variation mentioned. Now we will calculate the least significant difference. First we will mention the probability levels at 5% and 1%. Type here 0.05 in order to calculate LSD at 5% probability level. Now enter the 0.01, 1% probability level. Now we will enter the formula according to this. Type is equal to, now type TINV, double click on TINV, probability. Now mention the cell where the probability is mentioned, 5% level. First we will calculate according to 5% level of probability. Now enter the comma and then degree of freedom, that is 15. Here we want to fix this cell or fix this reference for future use. To fix this cell, place the $sign here. Before and after the alphabet. Close the bracket. Now multiply this and close this whole entry in order to represent this as one value, multiply by square root of 2 into error mean square divided by replication. Square root sqrt, double click, type 2, multiply by, type star, error mean square, here is error mean square, close the bracket, close this whole entry in one bracket, and divide by number of replications. Here is the number of replication, mention the cell. Now again close this whole entry in one bracket, we also want this cell and this cell fixed. Now we will place the dollar sign before and after the alphabet. This M7 is the error mean square, okay, and this Q2 is the replication. The formula is completed. Now we will press the enter to get the answer. 251. Now we will drag this value downward or copy this value, copy or place or paste it here. As you have seen that when we check this formula, everything is in place due to the dollar sign except for Q10, that is the LSD according to 5%. When we select this LSD at 1% probability level, click on formula bar, you will see this formula is shifted downward at the 1% level, and rest of the values are fixed. As you know, we have placed the dollar sign each and every cell except for this cell to fix the reference, and we do not need to enter the formula again for LSD at 1% probability level. Okay, when the CV is calculated from ANOVA, here is the CV. When the CV is calculated from the ANOVA, it helps us understand the relative variability and precision of the experiment for that trait being studied. Coefficient of variation is used as an index of experimental precision or reliability. It expresses the standard deviation as percentage of mean. If CV is less than 10%, the experiment is highly precise with very little environmental error. If CV is between 10 to 20%, the experiment is reasonably precise with moderate variation caused by environmental factors. If coefficient of variation is greater than 20%, the experiment is less precise, indicating higher experimental error or uncontrolled variability. It is to be noted that these ranges may vary depending on the trait. For example, yield data can tolerate higher coefficient of variations compared to the plant height. Furthermore, in Excel, the function of TINV uh, gives the inverse of students' T distribution. It means T inverse. In other words, it returned the T values such that the total area in both tails of the distribution equals the chosen significant level, that is alpha. Here is the alpha written. We calculate LSD only when the ANOVA results show significant F value, meaning the probability value is significant. Here we have got the non-significant result for genotypes, so there is no need to calculate the LSD for this trait. But I have done this for you in order to mention you the method that how to calculate the LSD in Excel. The LSD value here is LSD value at 5% level, and here is the LSD value at 1% level. It helps us determine whether the difference between two genotype means is statistically significant or not. If the if the difference between two genotypes, if the difference between these two genotypes or any two genotypes is equal to or greater than LSD value, those genotypes, then the LSD value, those genotypes are significantly different for that trait. If the difference is less than LSD value than two genotypes, then the two genotypes are considered statistically similar for that trait under study. So that's how you calculate and interpret the coefficient of variation and the least significant difference using ANOVA results. Thanks for watching, and I will see you in the next video. Take care.